DNA Repair. It's good to be back with you once again. Welcome to this lesson on DNA repair. We would like to alert listeners that it is advisable to study DNA replication before one goes on to this topic on DNA repair. In fact, the process of replication itself incorporates proofreading and error correction, which are related to DNA repair. One cannot emphasize too much the significance of DNA repair. DNA codes for life. It carries the information that defines a species and hence is responsible for all the characteristics and traits of a given organism. Hence, any alteration or injury, damage to it, can result in changes in the features of an organism or how it functions. Even a single base change can have these effects. Nature, therefore, has worked out several systems and mechanisms to ensure the fidelity of DNA, which can be defined as the property whereby a DNA molecule is not allowed to deviate from its original sequence. This definition is valid for haploid and diploid cells as well. In fact, the survival of a species can be at stake if its nucleotide sequence is not preserved intact. We can say that there is no other molecule whose integrity is as vital to a cell as DNA. Damage and changes in DNA can occur as early as replication up to the resting state of a cell, even when the molecule is inactive. We can suggest a few questions whose answers will make it easy to follow this theme of DNA repair. Let's consider the following questions, which we will then try to answer one by one. How can DNA be altered or damaged? What are the defects that occur as a result? Are there different mechanisms to take care of different kinds of alterations and damage? How do each one of them work? How can DNA be altered or damaged? Let us find out the answers to the first question that we have posed. How can DNA be altered or damaged? There are several mechanisms that can do this. Alterations can occur through base substitutions during replication. Further, the nature of the bases can cause base changes to result due to the inherent chemical instability of the bases or the N-glycosidic bond. Lastly, the action of certain chemicals and environmental agents can bring about damage and changes in DNA. These mechanisms are responsible for the occurrence of different types of defects. What are the defects that occur as a result? They are the following: incorrect bases, missing bases, altered bases, single strand breaks, double strand breaks, cross-linking. An incorrect base is a base that is not complementary to the template strand. It therefore does not have the appropriate hydrogen bond pairing with its complementary partner, and hence the Watson-Crick complementarity rules are violated. It can be a replication error that by chance is not corrected by the editing function of the DNA polymerase, or can be the result of a spontaneous loss of an amino group, deamination, thus converting cytosine to uracil or adenine to hypoxanthine, and thus the DNA molecule ends up with this defect. Missing bases come about through a process referred to as depurination, wherein the N-glycosidic bond of a purine nucleotide is spontaneously broken at physiological temperatures. It is true this happens at a very low rate. The absence of the purine base in the DNA molecule is equivalent to a non-informational site. Note, however, that the phosphodiester backbone of the molecule is intact. Such spontaneous depurination occurs at a rate of 10⁴ purines per day in a mammalian cell. Altered bases are bases that have been changed into comparatively different molecules. A variety of chemicals and physical phenomena can act as agents that do this. One example is ionizing radiation, like beta particles emitted by naturally occurring radioisotopes or X-rays too, can break purine and pyrimidine rings. Ultraviolet radiation can cause the formation of a variety of dimers between adjacent pyrimidines in a strand of DNA. Thymine dimers are the most prominent and can be seen in the figure. Following ultraviolet irradiation, thymine bases that are next to each other in a DNA strand are joined by formation of bonds as shown in the figure. The bonds formed are considerably shorter than the spacing between the planes of adjacent thymines, leaving the double-stranded structure of the DNA in the region distorted. Further, the shape of the thymine ring also changes, since the C double bond of each thymine is converted to a C single bond C in each cyclobutyl ring. As a result of the thymine dimers, the following significant effects occur. The DNA helix becomes distorted as the thymines are pulled towards each other as depicted in the figure, and hydrogen bonding to adenines in the opposing strand is significantly weakened, although possible because the hydrogen bonding groups are still present. Some chemical carcinogens, an example is those found in cigarette smoke, bond covalently with bases and bring about substantial distortion of the DNA helix. There are others that simply add a methyl group at an irregular position, such as the O position in guanine. Such changes do not interfere with replication, but during replication, the altered guanine is read as though it were an adenine. Single strand breaks occur when a phosphodiester bond in one of the DNA strands is broken. Phosphodiester bonds are susceptible to breakage by a variety of agents. Common chemicals among these are peroxides and metal ions such as ferric and copper. Ionizing radiation also causes strand breaks directly and indirectly by producing highly reactive free radicals in the water surrounding the DNA. Double strand breaks occur when a DNA molecule has a sufficiently large number of randomly located single strand breaks. It can then happen that two such breaks may be situated opposite one another, thus resulting in breakage of the double helix. Cross-linking involves a reaction in which a base on one strand links with a base on the opposite strand. Antibiotics, such as for example mitomycin C, and reagents like nitrite ions can form covalent linkages between a base in one strand and another base in the complementary DNA strand. During DNA replication, therefore, strand separation is hindered, and moreover, a local distortion of the helix at that point occurs. Cisplatinum and sorelin, activated by ultraviolet light, are also cross-linking agents. Their effect on inhibiting DNA replication has been used effectively in chemotherapy for cancer and in hyperproliferative skin diseases like psoriasis. Repair of incorrect bases. Let us now see the repair mechanisms that address the various damage situations of DNA. We have mentioned that prevention of errors when DNA is being replicated and copied is an integral feature of the polymerase enzymes involved. Thus, both DNA polymerase III and DNA polymerase I have editing functions to take care of this. The need to ensure that the sequence of DNA is preserved intact from one generation to another is further emphasized by the fact if the polymerase level of mismatch error prevention fails, then another system does exist for correcting the occasional error missed by the polymerases. This correction system is called mismatch repair. In this system, a pair of non-hydrogen bonded bases is recognized as incorrect. A polynucleotide segment containing this incorrect pair is excised from one strand, resulting in the removal of one member of the unmatched pair. The gap that is created is filled by DNA polymerase using this second chance to get it right. The final phosphodiester bond to seal the final gap is made by DNA ligase. The alert student will wonder how the system is able to distinguish which of the bases in the mismatched pair has the incorrect base. For this, it is important to identify the daughter strand, since it is this strand that will contain the incorrect base. The location of particular adenines in the sequence GATC provides the clue. These bases carry methyl groups not found in the DNA elsewhere. Methylation of the adenines in this sequence is associated with replication occurring in the daughter DNA strands. However, while parental strands are fully methylated, the newly synthesized daughter strands near the replication fork are not yet methylated. The mismatch repair system recognizes the degree of methylation of each strand, and when a mismatch is found, it preferentially excises nucleotides from the undermethylated strand, that is, from the daughter strand. Thus, the parent strand is always the template, enabling the repair system to correct such errors. Many types of cells use schemes other than adenine methylation to designate parent strands for mismatch repair. Another instance when incorrect base pairing happens is, as mentioned earlier, when cytosine loses an amino group by deamination and becomes uracil. In the second round of replication, this results in the replacement of a GC pair with an AU pair, which again, after the next round of replication, would become an AT pair. In order to prevent such mutations, a mechanism for replacing the unwanted U by a C has been evolved by nature. In the figure displayed here, the first step of this repair system shows the removal of the uracil by the enzyme uracil N-glycosylase. This enzyme cleaves the N-glycosidic bond and leaves the deoxyribose in the backbone. Then AP endonuclease, an enzyme that acts on apurinic or baseless sites in general, makes a single cut, freeing one end of the deoxyribose. This is followed by removal of the deoxyribose and several adjacent nucleotides, possibly through exonuclease activity of pole 1, after which pole 1 fills the gap with the correct nucleotides. This sequence, endonuclease, exonuclease, polymerase, is, by the way, an example of a general repair mechanism called Excision Repair, which we will talk about later. Also, note that the steps from AP endonuclease shown in the figure are also used in the repair of missing bases when depurination occurs. A specific glycosylase is also available to remove hypoxanthine, which results when adenine is deaminated. Yet another situation involves the base 5-methylcytosine found in many eukaryotes when it spontaneously deaminates. It then becomes the normal base thymine and mispairs with guanine. Again, this particular mismatch is recognized by an enzyme which will always remove the mispaired thymine rather than the guanine, so that the correct base pairing sequence is restored. The new cytosine can then be remethylated at a later time. Repair by direct reversal. The simplest mechanism for repair of altered bases is to restore them to normal without acting on the DNA molecule in a significant manner. In the case of the pyrimidine dimers formed in DNA by ultraviolet rays, this restoration is achieved in a very simple fashion. An enzyme called photolyase recognizes and binds to these dimers. It is activated by visible light and cleaves the dimers to yield the intact pyrimidines. Photolyase is found in many types of cells, but one must note that it is active only on pyrimidine dimers and in the presence of light in the 300 to 600 nanometer range. A methyltransferase that recognizes O6-methylguanine in DNA and removes the problematic methyl group is also an example of direct reversal. The enzyme attaches this methyl to one of its own cysteines. However, though the guanine is restored in the process, what is noteworthy is that the enzyme itself is inactivated. One could perceive this as an instance of how much nature is willing to pay to keep the DNA sequence intact and unaltered. Excision repair. The next repair scheme we take up for discussion is available very widely and is hence considered a ubiquitous repair mechanism. It is also one that can address a number of structural defects in DNA. The system is referred to as excision repair and involves a multi-step enzymatic process that has been studied and best understood in E. coli, in which it was originally discovered. This mechanism can repair pyrimidine dimers in the DNA in the absence of photolyase. Colloquially, another name for the scheme is cut and patch. The first step involves making a cut or incision. A repair endonuclease recognizes the distortion produced by a thymine dimer and makes a cut in the sugar phosphate backbone near the dimer. In E. coli, it involves the product of 3 genes, UVRA, UVRB, and UVRC, that act in concert to produce 2 incisions 12 to 13 nucleotides apart, so that the dimer or other lesion is contained between the 2 incisions. However, a single cut can adequately initiate the repair sequence. As shown in the figure, there is a 5' phosphate group on the side of the cut containing the dimer and a 3' hydroxyl group on the other side. The 3' hydroxyl group is recognized by polymerase I, which can then synthesize a new strand while displacing the DNA segment carrying the thymine dimer. This segment is then excised by the 5' to 3' exonuclease activity of pole 1. The final step is joining the newly synthesized segment to the original strand by DNA ligase. The student may note here that our insistence that a study of DNA replication should precede this lesson makes good sense. Some additional points about the excision repair system that are noteworthy are: it has been shown to preferentially repair pyrimidine dimers that are in certain essential regions of the genome, notably within expressed genes. In genes that are being actively transcribed, the repair is somehow targeted selectively to the strand that is being used as a template for messenger RNA synthesis. This special treatment is vital, since a pyrimidine dimer poses an absolute block to the process of transcription and could kill the cell quite directly if it prevented expression of an essential gene. The excision repair system requires many more enzymes in mammalian cells, as the chromatin structure is much more complex than in the simple E. coli. In fact, at least 9 or 10 genes are involved in the incision step. Xeroderma pigmentosum is a human genetic disease in which the victims are extremely sensitive to sunlight and further develop skin cancer after very short exposures to the sun. It involves a deficiency in one or another of the genes controlling the incision step of excision repair and a reduced ability to remove thymine dimers from DNA. Cocaine's syndrome is also a genetic disorder characterized by sensitivity to sunlight, but without the tendency to develop cancer. Interestingly, the defect in Cocaine's syndrome affects the preferential repair of expressed genes discussed above. Recombinational repair and translation synthesis. In spite of the availability of the excision repair systems for the removal of many of the thymine dimers and other lesions, its intervention may not be timely enough to avoid the effect of several dimers and their interference in various cellular processes. Such deleterious effects of some of these remaining dimers are eliminated by a process called recombinational repair and involves the system responsible for genetic exchange. We must first grasp how a thymine dimer stalls replication. In order to discuss the mechanism of recombinational repair, it is necessary to know the effect of a thymine dimer on DNA replication. When pole 3 arrives at a thymine dimer, the replication fork is stopped. A thymine dimer can still form hydrogen bonds with two adenines because the dimerization does not alter the groups involved in hydrogen bonding. But the helix introduces a distortion in the helix. So, when an adenine is added to the growing chain, pole 3 reacts to the distorted region as if a mispaired base had been added, and its editing function then removes the adenine. The cycle begins again. An adenine is added and then it is removed, with the net result that the polymerase is stalled at the site of the dimer. The same effect occurs if instead of a dimer, radiation or chemical damage resulted in formation of a base to which no nucleoside triphosphate could base pair. A cell in which DNA synthesis is permanently stalled cannot complete a round of replication and hence does not divide. In order to get DNA synthesis to be re-established, two basic approaches have been involved. They are post-dimer initiation and trans-dimer synthesis. The figure displayed on the screen illustrates how a cell could deal with a thymine dimer block. It bypasses it and initiates chain growth beyond the block, perhaps at the starting point for the next Okazaki fragment. Of course, this process results in large gaps in the daughter strands corresponding to each unexcised thymine dimer and cannot produce viable daughter cells, since these strands will give problems in continuing replication. However, a recombination mechanism called sister strand exchange comes to the rescue to ensure proper double-stranded molecules. In sister strand exchange, a single-stranded DNA segment free of any defects is excised from a good strand of the homologous DNA segment near the replication fork and somehow inserted into the gap created by excision of a thymine dimer as shown in the figure. Once again, as you may have guessed, pole 1 and DNA ligase act to join this inserted piece of adjacent regions, thus filling in the gap. The gap formed in the donor molecule by excision is also filled in completely by polymerase 1 and ligase. Through such exchange and gap filling for each thymine dimer, two complete daughter single strands can be formed, which can each serve as templates for synthesis of normal DNA at the next replication cycle. However, if two dimers in opposite strands are very near one another, no undamaged sister strand segments are available to be excised and the system fails. Recombinational repair is an important mechanism because it avoids delaying replication for the many hours that would be needed for excision repair to remove all thymine dimers. Besides, some kinds of damage cannot be eliminated by excision repair. For example, alterations that do not cause helix distortion but do stop DNA synthesis and can be addressed by this system. Since recombinational repair occurs after DNA replication as opposed to excision repair, it is often called post-replicational repair. Significantly, what is repaired is a daughter strand gap rather than a thymine dimer. There is evidence that thymine dimers and other helix distorting lesions block the progression of DNA polymerase, and yet it is clear that most types of cells, including mammalian cells, can tolerate large numbers of persisting lesions without resorting to recombinational strand exchange. Somehow, replication is able to overcome the lesions at the cost of an expected high error rate at those non-coding sites. Inducible responses. The DNA damage responses we have discussed are active all the time, that is, constitutive. But a few are inducible and are activated in response to signals like a blocked replication fork. One such very notable one is the Sos system. It has been characterized in E. coli. It is a complex regulatory scheme involving a Rec-A gene whose product, when activated by binding to single-stranded DNA at blocked replication forks, governs the expression of a dozen other genes. Some of these genes, which include UVRA and UVRB, are involved in excision repair, while others like UMUC and UMUD seem to be required to help replication bypass the offending lesion. Most lesions provide misinformation or no information to the polymerase enzyme at replication, and hence, though the system prevents termination of replication, it tolerates errors or mutations and so is called error-prone replication, and results in a higher than normal level of mutagenesis, though it provides a chance of survival. The Sos system was discovered through experiments which analyzed the survival of UV-irradiated bacteriophage lambda plated on E. coli. The survival of the UV-damaged phage was markedly enhanced if the bacterial host had also been irradiated with a low UV dose, though in this case a much higher percentage of mutants were obtained among the surviving phages. This phenomenon, termed UV reactivation, is now understood in terms of the Sos system. The low UV dose to the bacteria activates the Sos error-prone replication of the damaged phage and increases their survival. Instances of inducible cellular responses to other types of environmental stress, such as heat shock, oxidative DNA damage, or agents that produce O6-methylguanine, also exist. In the methylguanine case, an adaptive response in E. coli increases the level of the methyltransferase enzyme 100-fold when the cells are conditioned by growth in the presence of agents which produce this type of damage. Summary. We have discussed how a DNA molecule can be altered. The following ways have been listed: incorrect bases, altered bases, single-strand breaks, double-strand breaks, and cross-linking. We have discussed how each of these changes in DNA can occur and the agents involved. Most importantly, we have emphasized that DNA repair is required since the survival and preservation of the genetic traits of an organism calls for the integrity of its genetic material, the DNA. This DNA has to be replicated without any changes in its sequence. Physical and chemical agents, including ultraviolet radiation, can alter bases in DNA. Pyrimidine dimers occur when UV radiation causes two adjacent pyrimidines to become covalently linked, resulting in distortion in the DNA helix, which in turn affect the replication process. Another type of damage involves one or more breaks in the sugar phosphate backbone of the helix. Mismatch repair is used to replace an incorrect base. The incorrect base is excised and replaced. 4 mechanisms exist for the repair of thymine dimers. Photolyase is an enzyme that cleaves the cyclobutyl ring of the dimer when activated by visible light. The excision repair system makes a cut in the backbone on either side of the dimer, allowing the subsequent displacement and subsequent replacement of the strand containing the dimer. Recombinational repair involves post-dimer reinitiation of DNA synthesis and sister strand exchange and prevents long delays of replication. The Sos repair system also prevents delay of replication. This system allows for trans-dimer synthesis at the expense of a higher rate of base misincorporation by the replication enzymes.